Lysosomes and disease. Defects in a normal function of lysosomes often cause a variety of pathological conditions in the animals, including man. The importance of hydrogen peroxide in phagocytic killing of microorganisms is illustrated in humans by a X-linked recessive condition called a chronic granulomatous disease. A granuloma is a collection of dead and dying macrophages. The disease is a deficiency of the oxidase enzyme that produces hydrogen peroxide in the digestive vacuole. The macrophages and neutrophils of these individuals are unable to kill effectively even common bacteria, which may live and multiply inside the cell, eventually killing it instead. Such people are prone to all sorts of fungal and bacterial infections in spite of an immune system that is otherwise normal. Other types of defects in lysosomal function include a failure to fuse with the endocytic vacuole and failure to digest a certain classes of chemical substances due to lack of one or more critical enzymes. For example, fusion of lysosome and phagocytic vacuole is sometimes inhibited for reasons that are not understood. When the vacuole contains live bacteria responsible for tuberculosis or leprosy, or when it contains live toxoplasma, a common protozoan parasite contracted from raw meat or a cat feces. Lack of fusion permits these organisms to live within the phagocytic cells and often to destroy them and to establish active diseases. Algae that live symbiotically in protozoa seem to be similarly protected. The only clue how fusion might be avoided in these cases come from the observation that concanavalin A induced endocytosis also results in vacuoles that frequently fail to fuse with lysosomes. Concanavalin A or Con A is a lactine, the plant protein that behaves very much like antibodies in their ability to bind to specific chemical configurations such as alpha D mannose or alpha D glucose of a plasma membrane and thus cause a cellular adhesion in plant cells. Con A is found to cause activation of lymphocytes by cross-linking with receptors of lymphocyte plasma membrane. The latter can anchor to the underlying cytoskeleton. However, high dose of Con A immobilizes many receptors of lymphocyte and prevents activation. Fusion of phagocytic vacuoles with lysosomes and specific granules may also come too soon. If fusion occurs before the plasma membrane has closed over the forming vacuole, contents of the lysosomes and specific granules may regurgitate through the vacuole and into the extracellular space. Because of the destructive character of these enzymes, a local inflammation, that is redness, swelling, tenderness, etc. may result. This process, especially common with neutrophils, is important in localizing an infection and hence not always undesirable. However, it is probably also responsible for some of the degenerative joint changes in rheumatoid and gouty types of arthritis. The effectiveness of colchicines in the treatment of gout is probably due to its inhibition of lysosomes and granule movement and fusion, which are processes that involve microtubules. Another type of defect in a lysosomal function occurs if an essential hydrolase is missing. Because there are many hydrolases, there are many, more than 24 of these lysosomal storage diseases. They are almost always inherited as autosomal recessive traits because one good gene out of the two alleles inherited from the two parents is usually enough to prevent symptoms. The diseases have names like Tay-Sachs, Hunter's, Hurler's, Niemann-Pick and so on, because they were described long before the lysosomal functions was understood. Most of the diseases result in accumulation of undigested lipids or polysaccharides, which eventually fill the cell and thus compromise its function. The types of cell most affected will be determined by which enzymes is faulty, but the central nervous system is commonly affected, resulting in a progressive lack of body control and loss of a mental function, leading to a slow death.